Hello everyone, welcome back. This presentation we will continue with the discussion on glycogenesis. In the previous presentation we already saw that the glucose is the monomeric unit which is being used for the synthesis of glycogen in the process of glycogenesis. The glucose, it gets phosphorylated, it gets activated by phosphorylation to form glucose 1 phosphate through glucose 6 phosphate. The glucose 1 phosphate, for the conversion of this glucose 1 phosphate to glycogen, uh, glycogen synthase is required, uh, which actually helps in which employs actually the UDP glucose as the glucosyl donor. Okay, so the glucose UDP glucose is formed with the help of the UDP glucose phosphorylase. Glucose 1 phosphate, it utilizes UTP for the synthesis of UDP glucose, and this particular process, it is being catalyzed by the UDP glucose pyrophosphorylase. During this process, a pyrophosphate is being produced, which later will get hydrolyzed to produce two orthophosphates, and the energy trapped in the bond between them, it is being released, and that can be used for running up the whole reaction. Okay, now later what happens to the UDP glucose? The glucosyl group of the UDP glucose, it is transferred to a an acceptor molecule. This acceptor, it could be either a glycogen primer or it could be the Dolichol phosphate, depending upon which is the acceptor, whether it is a Dolichol phosphate that receives the glucose, or whether it is the glycogen primer that is going to receive the glucose. The pathways are two different pathways can be recognized. One is the glycogen primer pathway, which utilizes the glycogen primer and which is the most important pathway for glycogenesis. Another is the Dolichol phosphate pathway, which is mainly involved in the synthesis of oligosaccharides, which involves like glucose, mannose, and N-acetylglucosamine, etc. Okay, but the glycogen primer pathway, it is mainly for the synthesis of glycogen through the utilization of glucosyl residues. Okay, so here what happens is the UDP glucose, it is transferred in the glycogen primer pathway. The the glucosyl residue on the UDP glucose, it is transferred to the terminal glucose residue at the non-reducing end of a glycogen chain. Okay, very small glycogen chain, which is considered to be a glycogen primer, and this will result in the formation of a alpha 1,4 glycosidic bond between the carbon 1 of the new glucosyl residue and the fourth 4 hydroxyl. Here you can see this will be better. Okay, so this is the UDP glucose and this is the glycogen primer, and here you can see this is the glucosyl residue of the UDP glucose. It will be transferred on to the glycogen primer, and during this process what happens is a bond is formed between a glycosidic bond is formed between 1' carbon of the new sugar, here you can see, with the hydroxyl group on the fourth carbon of the non-reducing end of the glycogen chain. Okay, this is the glycogen chain and this is the non-reducing end. So what happens, there is a bond between formed between the fourth carbon of the non-reducing end of the glycogen chain and first carbon of the new sugar. Okay, and this will result in the formation release of water molecule and formation of a new glycosidic bond, and a new non-reducing end is formed. Okay, and the whole process, it is being catalyzed by the enzyme glycogen synthase, and this releases UDP. Now the glycogen synthase, it requires a primer, as already mentioned. It requires as a primer and 1,4 polyglucose chain. That is what the glycogen primer act as. Okay, and this glycogen primer has to be at least 4 glucose residue in length. Okay, and only in that condition it can act as a glycogen primer. Okay, so only when the chain is more than 4 glycosyl unit long, the glycogen synthase can catalyze the transfer of glucosyl residue from the UDP glucose. Okay, so once it finds a glycogen primer with at least 4 glucoside residues, the glucose is added onto the non-reducing end, 1,4 glycosidic bond is formed, and the chain length increases like this. Okay, it will result in an increase in chain length. Now what happens is when the chain length, the elongated glycogen chain, it becomes almost like 8 to 10 units long. another enzyme it comes into play. Actually, this is just a linear chain which is getting produced. But for the synthesis of glycogen, what is needed? A branching has to be developed. And this branching is done, cannot be done by the glycogen synthase. Instead, there is a new enzyme which comes into play for developing the what you call the bond, the branching, and that is a branching enzyme. Okay, the branching enzyme, it is otherwise referred as the amylo transglycolase or the glucan branching enzyme. Okay, here you can see, right, glucosyl alpha transferase enzyme. Now what they do is they will this particular enzyme, it will make a break, for example somewhere here. Okay, somewhere here we can see, so it will make a, yeah, it will make a break, it will break a alpha 1,4 glycosidic bond and separate a short stretch of glucosyl residues. And this short stretch which is separated, it will be added on to some other residue in the same chain or in some other chain, but here it is on the same chain. Okay, so here you can see it is 1 to 16 glucosyl residues are there, and somewhere just behind, just like after the 11th glucosyl residue or 10th glucosyl residue, a break has been made. Alpha 1,4 glycosidic bond is broken. Okay, and like the 11 to 16, right, these short stretch of glucosyl residues is separated from the linear chain, and it is being added onto the 7th sugar over here. Okay, it needn't be always the 7th, but here it is added onto the 7th one, and a new bond is formed. It is a alpha 1,6 bond. Okay, so the branching enzyme, it will break the alpha 1,4 glycosidic bond, separates a short stretch, a fragment of glucosyl residues from the linear chain, and then it reattaches to the chain through a alpha 1,6 glycosidic bond. So this creates a branching point over here. This is a branching point. Now what happens is there are two non-reducing ends here, one on this side and one on this side, on this end, right? Now the glycogen synthase, it can add glucosyl residues to both the ends, right? And then whenever a branching has to be made, new branching is done with the help of this glycogen branching enzyme, and there a new branch will be produced, a new branch, what you call the non-reducing end will be made, and the glycogen synthase can continue with its work. So it is actually glycogen synthase and glycogen branching enzyme, they will continue and they alternate their actions, resulting in the formation of a highly branched glycogen molecule, a glycogen polymer. Okay, and so branching, it makes the glycogen molecule soluble and more globular, and it creates more non-reducing ends for the action of the phosphorylase enzyme. Okay, so this is what happens. So this is the glycogen primer pathway where UDP glucose transfers glycosyl residue onto the non-reducing end of the glycogen chain. And here you can see the branching enzyme, how it works. It breaks a 1,4 glycosidic bond, breaks it, and then attaches it to some other, to a glycosyl residue on the chain, by way of alpha 1,6 glycosidic bond, which creates a branching point and a new non-reducing end. Okay, so this is what happens. Now we can see the Dolichol phosphate pathway. In the case of Dolichol phosphate pathway, the dolichols, they are long chain unsaturated isoprenoid alcohols. Okay, so it is phosphorylated dolichol, and here you can see this one is the isoprenoid. You can see almost 16 to 21 isoprenoids units may be there in a dolichol. That is almost 84 carbon atoms long, and they are usually found free or in phosphorylated. Dolichols are found free or in phosphorylated forms in the membranes of endoplasmic reticulum as well as in Golgi bodies. And the phosphorylated dolichol, it gets glycosylated at this particular point with the help of nucleoside diphosphate sugars. Okay, that is, for example, GDP sugars, right? So GDP sugar or GTP is required for transferring the sugar onto this one, right? Any nucleoside diphosphate, for example, it is in this one, it is GDP sugar. Okay, so sugar residue is transferred to the end of the growing chain of the molecule. So glucosyl units, it is transferred from UDP glucose to an intermediate acceptor, that is a dolichol phosphate, and the dolichol phosphate bound oligosaccharide is formed, and this oligosaccharide, it is then transferred onto a protein molecule with the help of a glycogen synthetase. Okay, and this forms a protein bound oligosaccharide, right? So it is a what you call protein plus the oligosaccharide, right? Okay,